everyone, my name is Myungjin Lee. I'm a bioinformatics scientist at Vaccine Research Center at NIH. In my poster, I'm going to talk about GLYCO-2.0, which is a web-based server to quantify glycan shielding of glycosylated proteins with improved data processing and computational speed. So before I go into the GLYCO 2.0, so if I talk about a little bit about the background, previously we developed an in silico tool that is called GLYCO, and GLYCO is from glycan coverage. So as the name says, GLYCO program is a tool to quantify or calculate the glycan coverage of glycoproteins. But the previous version of GLYCO is done by coding or programming, so we found that it would be great to make this program as a web server so that many people can easily use, even if they are not familiar with coding or programming. So if I go a little bit down, this is the link of the GLYCO 2.0 server. You can go and try. So motivation of the study, I brought some of the glycoproteins with some glycans. We wanted to develop a program to calculate the glycan density of glycoproteins and then get the value so that we can compare the glycan density between different glycoproteins, or within the structure, which part is the glycan high in density or which part is low in glycan. So below, I have the algorithm of GLYCO program. So the key algorithm of GLYCO is the same on both GLYCO 1.0 and 2.0. So basically the idea is to find out the glycan atoms around the surface area, a surface residue of the protein, and then check if the glycan atoms are actually giving the shielding effect to the surface atom, and then if it is actually giving the shielding effect, then we include that glycan atoms in the calculation, and once we count the number of these glycan atoms, it'll be our glycan density. So left one is the GLYCO 1.0 key algorithm, and then the right one is comparison between the previous version and then the current version. We changed a little bit of the algorithm from the numerical solution to the analytical solution. And then bottom is the GLYCO output. GLYCO can be applied to multi-frame as well, which is many different hundreds of PDBs coming from the molecular dynamic simulations. So with your glycoproteins, you can run GLYCO, and then you can find out the glycan density on the protein surface with a single PDB, or you can also use multi-frames so that you can get the idea of the flexibility of glycans during the molecular dynamic simulations. And then the right side, I brought many applications that was published with GLYCO program. Basically, as I showed you in the motivation of the study, after calculating the glycan density for these glycoproteins, we were able to find out the exact value of the glycan density for each structure, and then we were able to compare which one is the highest glycan density, which one is the lowest in glycan density. Below that, we also found that for the influenza HA subtype H3N2, it accumulates the glycan density at the head of the structure, as you see as time goes on, so that it tries to evade the immune response. So GLYCO has an ability to find out the glycan accumulation over time. Below that, another application of GLYCO is to find out the glycan hole, as you see in here, which is a really critical place in vaccine design, because normally antibodies are trying to bind the glycan hole area of the antigen. Additionally, GLYCO has an ability to calculate the glycan density just for the specific epitope, and then it can be related to the other antigenic properties such as antibody-antigen framework distance. That means when antibody tries to bind to the antigen, it has some distance between the main structure to the antibody itself. And each antibody has different distance, and we found that there's a high correlation between the distance and glycan density. So there are many utilizations and applications of GLYCO. So we found that it has a huge contribution to the vaccine design or immunology research. Thank you very much.